All right, you really can't understand what Mendel did and genetics without understanding probability, at least to some extent. Uh, and remember that Mendel was the first to apply probability statistical analysis to his results. And that's really the biggest reason that he discovered what he discovered. So when you look at a Punnett square like this, these letters that are on the outside of the Punnett square actually represent possible gametes. In other words, this parent having both a dominant allele for tall and a recessive allele for short, when those alleles get segregated into gametes during gametogenesis, gametes may be produced that have the dominant allele for tall, and gametes may be produced that have the recessive allele for short, because each gamete gets one allele from each gene from the parent. So if this was the male, these would be sperm. These would be possible sperm. And if this is the female, then these would be possible eggs. The percentages along with those letters, with those alleles, represents the probability that this parent might produce sperm with a dominant allele for tall or a sperm with a recessive allele for short. In other words, half this parent sperm would be expected to have the allele for tall, and half would be expected to have the allele for short. Same with the female with her eggs. So it's like flipping a coin. It's a 50-50 chance. Then, of course, as you know, the pairs of letters within the Punnett square represent possible offspring. So again, everything is possible. Possible gametes, possible offspring, because Punnett squares are used to simply predict what might happen, possibly. So likewise, the percentages along with these represent the possibility of having any one of these four combinations of these gametes. So there's a 25% chance that the dominant gamete from the father and the dominant gamete from the mother will come together to form this offspring, and so forth. And each is 25% because there are four possible combinations, and that's why there are four squares in a Punnett square, too. And there's an equal chance of each of those four possible combinations of gametes. So to put a definition on probability, it's the chance or likelihood of something happening or being the case. It's the possible outcome, but it's not absolute for sure. That is, unless the probability is 100%. So like I said, the function of Punnett squares, they are a tool for being able to predict. So in one word, that's what Punnett squares are for, is to try to predict the possible outcome of any particular mating between individuals. So of course, this represents Mendel's experiment going from the F1 generation to the F2 generation. And when he did this, when he actually crossed plants, no matter which character he was working with, it always ended up being the same. He always ended up getting results that were close to the predicted ratios. Not exactly. He may have gotten exactly, a few times, but for the most part, all he got were results that were close to what he predicted, which is good enough to support his hypotheses. The particulate hypothesis and the law of dominance or the principle of dominance and the law or principle of segregation. In other words, what he imagined was happening here in the way that offspring were being created from the parents genetically turned out to be right. His results supported his hypotheses. But they didn't have to be right on, they just had to be close enough. And the reason they weren't right on exactly what he predicted or what this would predict is just simply because of random chance. And that's all part of the whole idea of probability, is that it's affected by random chance. So as stated on the previous slide, probability can only predict the possible outcome. It cannot determine the actual outcome. And in genetics, results close to the predicted values are more likely with more offspring. In other words, the more offspring, and that's why Mendel, crossed hundreds and thousands of plants, is because he wanted to get enough offspring to be able to average the results and hopefully get something close to the truth, That's the way I refer to it anyways, close to the truth. So these predicted values can be considered the truth, but what actually happens may not always be the truth, but the more you do it and average those results together, the closer it should get to the truth or the predicted values. So analogy here between what we're talking about and flipping coins, but notice there's an analogy here between the coins and the Punnett square. We'll come back to that in a minute. But if you flip 20 pennies, like we have here, you would predict 10 or 50% or half should come up heads and half should come up tails. That's what you would predict. And if you count these up, you'll note that it is a 10 to 10 ratio. So this matches the prediction. But if you actually do this one time, it's likely that you won't get exactly 10 and 10, half and half. We would predict half and half, but in actuality, we really don't expect that to happen. If we were to do this just one time, I mean, I would put money on it not happening, actually. But I'm not a gambling man. But anyway, just by random chance, it cannot happen. It might, but it might not. So if this is just one trial, and this one came up 12, 12, 8, if you count them up. So this one trial didn't match the predicted values. But if you did this over and over and over again and averaged the results, then you would expect to get closer to a 10 to 10 or right on 10 to 10 eventually with enough trials. And that's not only important in probability, that's important when doing scientific experimentation. The more you repeat it, the more subjects you have in your experimental groups, the more you repeat the experiment and average your results, the closer you should get to the truth. So to ensure getting close to or exactly what you predict requires many flips or many offspring. So in this coin Punnett square analogy, the coins represent, a coin represents a heterozygous individual, a heterozygous parent, because a heterozygous parent has two different alleles, one dominant, one recessive, just like a coin has a heads and a tails. And there's a 50-50 chance that when you flip a coin, it's going to come up either heads or tails, just like there's a 50-50 chance that a parent is going to produce a gamete that has either a dominant allele or a recessive allele. So the analogy is a good one. So anytime you flip a coin, there's half a chance that it's going to come up heads. And so there's this concept in probability theory known as the multiplication rule, which states that for any two independent events, like flipping a coin, so flipping a coin, one coin is an independent event, the probability that both events will occur simultaneously, in other words, when you flip two coins, the chance of them both coming up heads, like we see here, is the product of their individual probabilities. In other words, 1/2 times 1/2 in this case, and that's 1/4. So the probability of flipping two heads with two coins at the same time is 1/4. And that equates to having an offspring that has two dominant alleles, like we see in the Punnett square. And that's the same for all these possible combinations, because all the gametes, there's a half a chance, 50%, 0.5 chance of getting any one of these gametes. And so when they come together in the Punnett square, the multiplication rule comes out with 1/4 for each one. And that kind of explains why there are 4 squares in a Punnett square, because there's a 1/4 chance of getting any one of these combinations of alleles from the parents, from these heterozygous parents. Each possible combination has a 1 in 4 probability. You'll want to note, though, that these two have the same genotype, really. Even though they do represent two different combinations, possible combinations, they have the same genotype. So there's a 1/4 chance of getting a homozygous dominant, there's a 1/4 chance of getting a homozygous recessive, but there's half a chance, 1/4 plus 1/4. That's the addition rule that explains the probability of getting a heterozygote, because there are two possible combinations that result in heterozygotes. All right, so here we're going to take a look at a word problem, a genetics word problem, and you solve these word problems just like you would in math class, a word problem in math. So in pea plants, round seeds are dominant over wrinkled. So you want to read and you want to highlight or underline or mark somehow things that are important. And there's a procedure you want to follow too. So round seeds are dominant over wrinkled. That's an important piece of information. And what you'd want to do here is come up with a symbol to represent round, the allele for round, and a symbol for the allele for wrinkled. And just by convention, what we usually do is we choose the first letter of the dominant trait to be symbol. In this case, round is dominant over wrinkled, so we would go with R. And round would then be a capital R, an uppercase R. Wrinkled then wouldn't be W, it would be a lowercase r. So we're being asked here to predict the genotypic and phenotypic ratios. So that's important information. That tells us what it is that we're being asked to come up with. So predict the genotypic and phenotypic ratios of a cross between one homozygous round plant and one homozygous wrinkled plant. And if you're paying attention, you might recognize that as being a parental cross, one of Mendel's parental crosses. As you read through, you would want to mark everything. So um, we're after the ratios. These are the symbols. The genotypes of the parents, you would also want to indicate or write down. So the homozygous round would be capital R, and the homozygous wrinkled would be lowercase r, lowercase r. And then it's a good idea to write it as a cross. In other words, capital R times lowercase r, lowercase r. Remember, a multiplication sign in genetics means a cross. So we're crossing these two individuals. And again, that's the parental generation, the P generation. Then, of course, we can set up and solve the Punnett square. And you already know what's going to happen because you're familiar with Mendel's experimentation. But just for the exercise here, let's go ahead and do this. So here's the homozygous dominant parent, the homozygous recessive parent, and then, of course, within the squares, there's the same combination. So all of them are going to be heterozygous in the F1 generation, and that's what Mendel saw too. And speaking of probability, the probability in this case is 100% of getting all heterozygous offspring, unless there's a mutation. Now, so even though it's 100% probability, there are always things that can happen, almost always things that can happen to not get exactly that predicted value. So now that we have the Punnett square, we can go after what it is that we're being asked to find. So the genotypic and phenotypic ratios. And again, you want to just kind of follow this procedure. I mean, you already know what the genotypic and phenotypic ratios are, I'm sure, but this is the procedure that I would recommend you follow when you're doing a more complex problem. This one's simple, but the next thing you do and I abbreviate genotypic ratio, G ratio here. So for the genotypic ratio, you want to list all the possible genotypes. And I'm not talking about the possible genotypes within the Punnett square. I mean all possible combinations of the alleles in the cross. So there's a possibility of getting big R, there's a possibility of big R, and there's a possibility of little r. In other words, homozygous dominant, heterozygous, homozygous recessive. And then you count up how many there are of each. And in this case, there's 0 homozygous dominant, there are 4 heterozygous, and there is 0 homozygous recessive. And then you would write that as a ratio. In other words, the numbers in that order, homozygous dominant, heterozygous, homozygous recessive, 0, 4, 0, separated by colons. And you can also describe it in words and just say they're all heterozygous. You do the same thing for the phenotypic ratio, and it turns out that homozygous dominant and heterozygous are round, would end up being a round individual phenotypically, and there are 4 of those. They're all heterozygous. And then for wrinkled, count them up, there are none, so that's 0. And so likewise, the ratio would be 4 to 0, but it's always in that order, dominant to recessive. So in this case, round to wrinkled, 4 to 0. Or you can say they're all round, if you're going to describe it in words. Here's another one in pea plants. Round seeds are dominant over wrinkled, so it's the same gene, the same character in the same gene. Find the genotypic and phenotypic ratios of a cross between two heterozygous plants, and hopefully you recognize this as being an F1 to F2 cross. So we're taking one of those offspring from the F1 generation and either self-pollinating it or cross-pollinating it with another F1 plant. Either way, we're going to end up with the same thing. So round seeds and wrinkled seeds, both parents are heterozygous, so big R, little r for both of them. And they represent the F1. We go ahead and create a Punnett square, place the parents on the outside of the Punnett square, and remember these are possible gametes produced from those parents. When the gametes come together at fertilization, they produce these possible offspring. There are 4 possible combinations. If we calculate the genotypic and phenotypic ratios, list the possible genotypes, count them up. In this case, there are representatives of each. So we've got 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So we write that as a ratio. That's a 1 to 2 to 1 genotypic ratio. And whenever you have a monohybrid cross like this, you'll always get a 1 to 2 to 1 genotypic ratio as long as we're dealing with complete dominance. So as long, and that's what all the characters that Mendel dealt with showed complete dominance. None of them showed incomplete dominance. So you will always get a 1 to 2 to 1 genotypic ratio in a monohybrid cross that shows complete dominance. Same for the phenotypic ratio. 3 round, 1 wrinkled. A 3 to 1 phenotypic ratio. You will always get a 3 to 1 ratio in a monohybrid cross that shows complete dominance. So you can take those to the bank. You can just memorize them. You don't have to do the Punnett square. If you know you've got a monohybrid cross, don't bother doing this. Just remember, it's going to be a 1 to 2 to 1 genotypic ratio, and it's going to be a 3 to 1 phenotypic ratio. However, if I ask you to show work, I will expect to see the Punnett square. As a matter of fact, if I ask you to show work, if I give you a genetics problem like this, I expect to see everything that I have here show work to get full credit. All right, this one's a little different. And what this has to do with is what's known as a test cross. A test cross is a way of finding out the genotype of an individual that has the dominant phenotype, but you don't know what the genotype is. So you've got an individual that has the dominant phenotype. In this case, that would be yellow peas, but you don't know if it's heterozygous yellow or homozygous dominant yellow. So what you need to do is actually do an experiment to find out. And what you do is you cross that individual with an individual of known genotype. And the only individuals that you know their genotype are the ones that are homozygous recessive. In this case, that would be green. So you would take that yellow pea plant, that plant that has yellow peas, and cross it with a plant that has green peas, because you know the genotype of that green pea plant. And that's called a test cross. Now there are 2 possible results from that, depending on the genotype of the yellow pea plant. If the yellow pea plant is homozygous dominant, then you're going to get a different outcome than if the yellow pea plant is heterozygous. So what this is asking you to do is show the 2 Punnett squares that represent those 2 possibilities. So again, you want to read it, you want to pick out the important information, decide what you're going to use for symbols. And Y's are kind of weird for symbol, or not weird, just kind of difficult to use for symbols, because you have to make very sure that you show the difference between uppercase Y and a lowercase y. So I tried to do that as best I could in this problem. So show the 2 possible outcomes of this test cross for this trait. So it's an experiment. I want to emphasize to you that what you would actually do this, you would actually carry out this breeding experiment to find out. So the question is the yellow pea plant homozygous dominant or heterozygous? We don't know. You can't tell by looking. It's a yellow pea plant. Can't see the genes. So if homozygous dominant, then we would expect the offspring to all be yellow. Okay, so this is the possible outcome. Here again is the homozygous recessive, the green plant that we're going to mate the yellow plant with, because we know the genotype of the green plant, it's homozygous recessive. So that's the whole idea. You mate the unknown plant with a known plant, and then you should be able to predict the possible offspring if that unknown plant is homozygous dominant. All the offspring would be yellow. So all yellow. If we write that out as a hypothesis, then if the individual of unknown genotype is homozygous dominant, yellow, and we cross it with an individual of known genotype, homozygous recessive, green, then all the offspring will be yellow. But it's an actual experiment. If the individual is heterozygous, then the Punnett square would look like this. We're doing the same thing, crossing the heterozygous, though, in this case, with the homozygous recessive, the individual of known genotype. And these would be the results. In other words, half would be yellow and half would be green. And that would tell us that the individual is not homozygous dominant, it's heterozygous, if there are any green plants at all in the offspring. So it would be a 2 to 2 ratio, yellow to green. And here's the hypothesis for that. If the individual of unknown genotype is heterozygous yellow, and we cross it with an individual of known genotype, homozygous recessive green, then half the offspring will be green. All right, so we would hypothesize these two predicted outcomes and then actually carry out the experiment and see what the offspring were. And if there were any green offspring at all, then we'd know that the individual of unknown genotype is actually heterozygous. If not, then we would know that they were homozygous dominant. That's a test cross. And obviously you can't do this with people, but you sure can with pea plants, and you sure can with fruit flies, and all kinds of other animals or other organisms. So we're going to sum up what Mendel has discovered up to this point. So Mendel's observations and discovery so far: one phenotype in the F1 and two in the F2. In other words, those results, that there was only one phenotype in the F1 generation. In other words, they were all heterozygous, right? So they all showed only one phenotype. And that there were two in the F2. In other words, the recessive trait reappeared, is what that's saying. That's what led him to conclude that the particulate hypothesis, or the gene idea, is the correct one, not the blending hypothesis. So these results supported the particulate hypothesis, that there are genes, there are particles, there are alleles that pass from parents to offspring. It also allowed him to conclude that the principle of dominance, it supported the principle of dominance, that one trait dominates over the other. Masks it. One trait masks the other is a good way to think about that. And his observation that the actual results fit the predicted ratios allowed him to conclude that the principle of segregation was correct. So he hypothesized that the alleles segregate in the production of gametes, and that's what we're seeing here. Each gamete only gets one allele. So the alleles segregate in the production of gametes from the parent, and that's true. They do. And how that happens is through the process of meiosis, not mitosis, but meiosis. Okay, so I wanted to stop and summarize here what we've discovered so far, or what Mendel discovered so far. But there's one more thing that Mendel is going to discover, and that's the principle of independent assortment. And in order to discover that, he had to do a dihybrid cross. And that's what we'll take a look at next time.